Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. The emergence of life on Earth is one of the most interesting questions for modern science, and the lab of Dr. Jack Szostak at Harvard University is one of the major centers of that research. Recently, Dr. Alonso Ricardo from the Szostak Lab recorded a program for Science for the Public on the whole sweep of this research. In his presentation, Dr. Ricardo mentioned the work of two Szostak Lab researchers that you will see on this program. Dr. Matthew Powner will discuss his work on the emergence of nucleotides in RNA, and Itay Budin discusses his work on the earliest cell membrane.
Hi, I'm Matthew Powner and I'm from the UK. I am going to talk to you today a little bit about some experiments we've done towards the origin of pyrimidine nucleotides, two of the four nucleic acids that make up RNA. So RNA is a vital part of molecular machinery, the molecules which allow life to be what we know it is today. RNA is key in the process of taking genetic information and translating it into usable protein compounds, enzymes, the functional molecules of biochemistry. One of the key questions to be answered formally by chemistry is how these RNA molecules, which are key to life as we know it, first came to be on the early Earth before life existed itself. For 60 years or more, people have been trying to put together the fundamental organic chemicals which are known to be able to be formed in simple environments by environmental chemistry, high energy irradiations and spark discharges such as lightning going through simple gases can create more complex molecules. These molecules are high energy, and it's known the energy in these molecules can be used to build more and more complex molecules. But for 60 years, people haven't been able to put together these simple organic chemicals with high energy and bring them together in such a way as to build ribonucleotides. So what we've done is had a new look at these same chemical species, and we've tried to put them together in new and different ways to what's been tried before to get the same end product that nobody else has been able to. So one of the major problems with nucleotide synthesis in an abiotic sense, the way we're looking at it, was a particular bond which is very difficult to form. This to chemists is known as the glycosidic bond. For years people have tried to take what looks like one of the key components of a ribonucleotide, which is ribose, the sugar central portion of the molecule, and to condense this with the nuclear-based portion. The nuclear-based portion is the part which allows it to transfer information from one generation to another through hydrogen bonding. But this condensation works very poorly with two of the nucleotides, the purine nucleotides, and doesn't work at all with the pyrimidine nucleotides. So to overcome this problem, we disconnected the molecule. We took it apart in a theoretical sense, which is the way chemists work with molecules. If you want to build a big molecule, you work backwards from that big structure and take off smaller structures that you might be able to put on at a later stage. And we call this retrosynthetic analysis. Our retrosynthetic analysis disconnected the molecule in a novel new way by breaking the sugar in half at the start of the disconnection. So instead of taking preformed sugar and trying to attach a nucleobase, we took what was in essence half nucleobase, half sugar, and the other half of the sugar, and connected these two molecules together before elaborating the rest of the nucleobase, and then introducing phosphate to finally deliver the nucleotides. I think the key is each of the components used is what we would call prebiotically plausible. So they're simple molecules, like 2-3 carbon maximum. All of the chemistry takes place in water. It's all aqueous chemistry, which is relatively unusual for organic chemistry. Most, if not nearly all, organic chemistry is done in organic solvents, not water. So each step is plausible, each step is high yielding. We're getting quite clean products at the end, because after the nucleotides have been built, there's a stage where we've shown that by irradiating them with UV light, those which are required and those which are the ones we're aiming to build are kind of self-protecting. The intrinsic structure of the molecule protects it from UV light, whereas all the other side products are not protected. Therefore, they're destroyed by UV light. So there's kind of a purification step at the end, which also happens to convert cytidine to uridine, so furnishing the second of the 4 nucleotides. And from the perspective of it being one complete synthesis or prebiotic route, each step is compatible with the next. So throughout the chemistry, phosphate, for example, is used to catalyze the chemistry, and it can be there present throughout the whole series of reactions, and in each reaction it's present. It actually improves the chemistry until the second to last step, where it's actually then incorporated into the molecule itself, phosphorylated, to give a nucleotide, which needs to include phosphate. So one of the things of the chemistry is all the individual parts come together to make one route which is supported by all the other parts. So each part supports the next. The chemistry is the same type of chemistry, it takes place, similar pHs. Phosphate helps each step, is required in one of the last steps. So I mean, it all supports the rest of the scheme. Each part is supportive of all other parts. The ones we did were the ones that couldn't be done.
Okay.
The ones that hadn't been seen in any form of yield. The other two, well, one of the other two has been made in very, very low yield. I personally think there will be better chemistry to make the other two as well, but we haven't as yet found it. We'd love to find it. And I think part of the whole process is to discover all 4 nucleotide syntheses, and then again there'll be feedback loops. So you can think about when you found syntheses, good syntheses of the purine nucleotides, how do they how would they interact with the pyrimidine syntheses if for example they were running side by side. So the first step is obviously to try and make individuals, and then you build up. Can you put it all together? Does all this chemistry work together? And then you've got something which corroborates your results. If you can corroborate the pyrimidine syntheses with purine syntheses of a similar nature, then you have further evidence that these could have been something important to the origin of life. The specific chemistry of the purine synthesis that the pyrimidine synthesis that we've published is all chemistry done in water, barring the phosphorylation, which occurs by drying down a solution of water, so heating water or evaporating it. All of it occurs at very mild conditions, so neutral conditions. Doesn't require any strong alkaline conditions you might expect at hydrothermal vents. It doesn't require any huge temperatures. Phosphorylation was done at a reasonably high temperature, but only it needs like 80 degrees, doesn't need hydrothermal temperatures. So I think the chemistry itself will work best in mild conditions. Strongly alkaline or acidic conditions will take the chemistry down other paths, because very harsh conditions like very strong alkali solutions will have all kinds of effects on chemistry. But say chemistry building nucleotides couldn't happen in other environments would be untrue. I think you could take this chemistry to other environments. But the important point is you don't need to. You don't need hydrothermal vents to generate hugely alkaline conditions to build sugars. We don't need that. We can put together the carbohydrate by simple chemistry in aqueous solution at neutral pH. We don't need hugely alkaline conditions. So there's no suggestion that there wouldn't have been these conditions on early Earth. Of course there would have been varied conditions across the Earth. It will be different in different locations. The key point of our chemistry is you don't need anything special. You need these simple molecules in neutral water, which is fairly ubiquitous across the whole surface of the Earth. People have endeavoured to look for more vigorous and strong conditions to try and do chemistry that they couldn't access before, because they couldn't access these molecules under simple neutral conditions. So I think the question is, do you need to move to them unusual locations to do this chemistry? You don't need extremes of temperature, you don't need extremes of pH, you don't need extremes to do this chemistry. It works well under very simple conditions, very natural conditions that you'd expect to find everywhere. Yeah, well, I think any discovery that shows people they can do things they thought they couldn't gives everybody else impetus to have a go as well, which only leads to more success.
Yes.
When I was young I should say, what I wanted to be was an engineer. I wanted to build things. I went to study engineering at university, but wasn't happy with what I was doing because it wasn't as hands-on and there wasn't as much kind of construction almost involved as how I'd imagined it. And so I kind of looked for a new course, a different thing to study, and I found chemistry, where you build molecules, specifically organic chemistry. And I kind of got hooked on organic chemistry and the ways you can use molecules to control themselves to build what are essentially specific architectures at this molecular scale. And then I became more and more interested in organic chemistry and predominantly studied that at university for 4 years, and then went for a short while to work with AstraZeneca as a medicinal chemist before deciding to go back to study a PhD in organic chemistry, where I studied with John Sutherland, who I did this work with. And I was looking for something novel, something different, something unlike what anybody else was doing. And John offered me this opportunity to study the potential origins of nucleotides and how you could use the intrinsic properties of these small molecules in what would be a prebiotic environment to build up more complex structures in a specific way. So there was a specific target and there was these specific molecules that you knew you had to use, and could you get from A to B? And I just loved the idea of doing that, and I've been doing it ever since. And it's frustrating but fun.
I'm Itay Budin. I'm a graduate student in Szostak's lab, and today we'll be talking about models for membranes, lipid membranes during early life, and how we try to recreate those structures in the laboratory. So all modern cells are encapsulated with membrane structures, and these are lipid membrane structures, what biologists would call a phospholipid membrane. And we think that similar structures would have been necessary very early on during the origin of life. So a lot of the work we've done is try to look at chemically simpler molecules that could have been around on the early Earth, but display sort of similar properties that self-assemble into very similar structures that could have served an analogous role for the first cells. When discussing questions of the origin of life, there's a couple of challenges, one of which is conceptually, you have to start thinking about how systems, which, biological systems are very, very complex, how they can be simplified into many, many fewer components. And the other thing is you have to think about a very fundamental level about the molecules involved and how they can behave and act independently of any sort of complex machinery that you see in biology today, and yet serve those same functions. So what I'm taking is a mixture of lipids. So these are fat-like molecules, and these are particular molecules that we think could have been available on the early Earth. So similar kinds of molecules have been found on things like meteorites, and we have sort of defined chemical pathways by which purely environmental processes can generate very similar species. So we're just taking a small amount of these lipids, and you see they're basically a clear oil, as such. And then what we want to do is add just some buffer. And by what I mean by buffer is a water solution in which the pH, sort of the acidity of it, is controlled. So what we're going to show is the self-assembly of simple fatty acid membranes. And this process turns out to be dependent on the pH of the environment. So you require a slightly basic pH for this process to occur. And if you have that, and if you have sufficient concentrations of the lipids involved, membrane self-assembly occurs spontaneously. So we start with our oil, we add our buffer, and then we just mix gently. As you can see, we result in a solution that is turbid and getting progressively more turbid. And what that signifies is that there's large nanometer to micron sized membrane structures that have self-assembled just in the time course of these few seconds. And these basically scatter light. So your solution becomes turbid, similar to, let's say a milk solution in which globules of fat and protein do a similar function. And so these are the kind of membranes that we think could have self-assembled on the early Earth by sort of similar processes. And a lot of the effort we've done in our lab is to look at these structures and ask what are their sort of physical properties. How do those physical properties differ from those of modern cell membranes? And what does that mean about the function of the first cells, potentially? So just a very simple experiment, but one that tells us a lot about the sort of processes that could have occurred very early on during life. So you really need two things. You need to have the right chemical components. So in this case, we're talking about a distinct class of lipids, these fatty acids. That's important, and you need them to be in sufficient quantities, right? In that self-assembly can't occur in very, very dilute solutions. You need relatively concentrated solutions for self-assembly to occur favorably. The second thing you need is the right chemical environment. So for example, in this experiment, we're tightly controlling the pH of the solution. We have a slightly basic pH solution, which is a requirement for these bilayers to self-assemble. If we had an acidic solution, you'd get basically oil droplets in water, similar to a salad dressing. If you had too basic of a solution, you wouldn't get bilayer structures, and you get a more soluble form, a micelle aggregate form, which also could not serve as a membrane. So these are the kind of constraints that we think about for the origin of life. And these kinds of experiments we do in the lab help us define what these constraints are and what that can tell us about the environment that was needed 4 billion years ago.